You are helping underwrite women's health initiatives like free mammograms and cancer screening through Project Pink, and also helping supply needed equipment for women's services at our hospital. So thanks for your support of this event. We're also, thank you. We're also appreciative of our great partners at UCSF Medical Center who are joining us again this year. And today, Drs. Cow and Miller will be sharing their expertise on memory, a subject which is especially important to all of us as women, since we are more vulnerable to memory problems than men. Perhaps they'll shed some light on why that is, I hope. Before turning the program over to John Henley, the CEO of Sonoma Valley Hospital, we also would like to give a special thanks to the hardworking committee who helped put this symposium together. So thank you, Prima Bayan, Lindsay Bennett, Kimberly Blattner, Suzanne Brangham over here, Leslie Conway, Susan Idell, and Judy Vadaz. Thanks, committee. And now, please welcome our hospital's leader, John Henley.
I always have to adjust the mic. Thank you. And thank you to Cherie and Joan and Marcia and the committee. Their support, both their time and their resources, have been incredibly impactful to Sonoma Valley Hospital. So thank you to the group for all that you do. I'd also like to recognize our host, Hannah Boys Center. The center provides vital services within our health network in behavioral health, both with their residential program and their soon-to-be launched outpatient program. I'm really excited about the hub that's going to be coming online in September. They will be providing outpatient services to all youth in the Valley and their families. So really looking forward to that. So I'm excited to be here in person. 2 years of virtual was great, but it's so much better to be here in person and to learn a little bit about brain health as we age and as women age. Our partnership with UCSF is foundational to our growth. They have provided this forum for us to learn. Dr. Josephson's support through the Department of Neurology of our best-in-class stroke program at the hospital has been phenomenal. And there's so much more to come, from specialists to strategists to their support of our infrastructure. They have helped Sonoma Valley Hospital go from a great hospital to an exceptional hospital. And I can only imagine what we're going to do over the next 5 to 10 years with that relationship. With that, I will introduce our moderator, Dr. Andrew Josephson, a graduate of my alma mater, Washington University in St. Louis, faculty and the chair of the Department of Neurology within the UCSF Weill Institute for Neurosciences. Dr. Josephson.
Well, thanks, everybody. We are all just delighted to be here, and I can't believe this many people showed up. Thank you so much for being here. This is fantastic. This partnership, as John has said, this is a very deep partnership between UCSF and Sonoma. I think many of you know this, but at least if I can just add a little bit to what John said on the neurology side, beginning now 3 years ago, our neurology department provides all neurologic services in the hospital 24 hours a day, 365 days a year. In that way, if somebody has a terrible thing happen to them, like a stroke or a seizure or anything else that they have to come to the emergency department, rather than having to get in an ambulance or a helicopter and fly somewhere really far from home, we come here and give you the same neurologic care than if you were at our place in San Francisco. And I think that's been, for us, just a wonderful partnership. It's exciting to take care of folks, and to be able to bring this kind of quality care to you has been something that's been very meaningful to us. In addition, these wonderful seminars that we've had, I think, have been a highlight. I want to thank Sherry, Joan, the entire committee for inviting us once again. And today we're going to talk about what has got to be one of the more scary but also exciting areas of neuroscience and thinking about memory loss, memory disorders. I have brought with me two of the greatest superstars in the world to talk, not just at UCSF, but these are world-renowned folks. And what we're going to do is we're going to have two relatively brief talks that I think you're going to find very engaging, and then have a Q&A session at the end that I'm going to moderate. You all got a, I think, branded notebook. That's great. Thanks, Dave, for putting that together. Those are not only to take notes, but to write down questions. So if you have questions as we go, please, let's get a bunch of them. Feel free to write them down, and then we'll have people pass them to the aisles, and they'll bring up, and then I'll do a little Q&A for the second half of our program. So it should be a lot of fun, but this is meant to be really interactive. So let's make sure we try to do that. Let me introduce our two speakers. So going second will be Dr. Amy Gao over there. Amy is a physician scientist in our department, a professor of neurology. Amy actually runs the training program at UCSF for all the medical students who want to do what she does, which is to be an outstanding clinician and at the same time a world-class scientist. So Amy will probably be way too modest about her work, but she is one of these people who not only is a great dementia doctor, but is in a laboratory using animal models to understand how these diseases work and to develop cures for these disorders. So we're really excited to have Amy here. She's going to talk second. And first off is going to be Dr. Bruce Miller to my right. Bruce is the head of our Memory and Aging Center. He's someone who many of you may have, just like Amy, heard of around town. But Bruce has given us some of the most fundamental insights into a variety of neurodegenerative disorders, including one that we're not going to spend as much time on today, called frontotemporal dementia, maybe the second most common cause of dementia after Alzheimer's disease. Bruce, we could go on and on, but I'm not gonna because he'll get mad at me, but a world-class clinician, a wonderful researcher, and has trained multiple now generations of behavioral neuroscientists who care for patients with these disorders. And like Amy and myself, who trained, both of us trained with Bruce, are sort of working hard to develop cures for these terrible diseases. So I'm going to turn it over to Bruce Miller. Thanks so much for being here. Keep those questions coming. Really appreciate it, everybody.
Let's see, I have some slides. I'm so grateful to be here today. When I was a fellow at UCLA in 1983, a woman named Dorothy Kirsten, a great opera singer, asked my teacher, Frank Benson, if he would help her set up an Alzheimer's program to fund great scientists. And Dorothy, who worked with women's groups in an extraordinary way, and I began to work together in 1984. And so it feels very good speaking to a women's group like this. Huge thanks to Joan Weil for this and the way she and Sandy are changing the world in such a great way. But I think we know women are the drivers in this space. They suffer the most as caregivers. And I think women scientists like Amy are going to find cures. I really believe that. And not in the far off future, but in the near future. And I hope I translate some of my enthusiasm about that. So I'm going to talk a little bit about the prevalence and things, really emphasize, which I think is what you care about, things that you can do in your own life to prevent ever getting sick. And I think many of you are doing many of these things, but I just want to talk about how much we've learned in terms of lifestyle about prevention of brain disease. It's no surprise how we live our lives would have an influence on our brain. But I think the data is now there. I'm going to talk about cognitive screening. If you're worried, what do we do? A little bit about dementia versus Alzheimer's disease. Some of the biomarkers that we now have that just strip away the myth that you can't diagnose Alzheimer's until after you've died. That's wrong. And this is really exciting in terms of new therapies. And then a little bit about treatment. So this is a massive healthcare problem. Here are some of the statistics. It is without a doubt the most costly disease in the United States. Probably $290 billion a year of our health budget goes to dementia care. So this is also a little bit of an epidemic. So dementia is increasing, and that means that if we don't find treatments for this, both in lifestyle, but also in medicines, we as a society will be overwhelmed by the degenerative brain diseases that people tend to suffer more as they get older. So this is, I didn't believe it at first, but this is something that I think increasingly has come to our attention, and that is that modifying 12, I'm going to list 14, risk factors might prevent or delay up to 40% of all dementias. When I read that figure, I just couldn't believe it. But the more we bear down on this, the more we realize that these things that we do with our lifestyle change the chemistry in the brain, and do things that scientists are also trying to do with drugs. So they go hand in hand, I think. Here are some of the risk factors. Not going to read these off, but I just point out that these all increase your risk for developing a degenerative disease of the brain when you get older. And they range from things like low education, incredible story about low education. People we've learned from one of our fellows, Elisa Rezende, who are between 45 and 60 and healthy, if they haven't learned how to read, their hippocampus, the memory area in the brain, is smaller than people who have. How does that translate in your own lives? I think it's important, and I think more and more we realize that people who are socially isolated, depressed, not cognitively or physically active, fall prey in very disproportionate ways to all the degenerative diseases. I've listed air pollution here, which increasingly is an issue, particularly in third world. But I think we now know from animal models that air pollution, even early in life, is injurious to brain systems, and if we age, eventually will cause cognitive disorders. So these are things that are so important for a society, that a society can change, but also you can change in your own lifestyles. This is about physical exercise, and it just points out that in our healthy aging cohort, which I hope some of you will join, we have learned that the super agers, the people that do better than their peers, almost all of them are exercisers. You're going to ask me how much exercise, and I think we don't know. Is there too much exercise? I think so, probably. But Andy doesn't believe it. Anyways, because he is in constant motion. There's nobody, and I'll come back to it, but he also has another risk factor for neurodegenerative disease. So I'll come back to it. But I think we have learned that people who are moving, who are exercising probably 5 to 6 times a week, at least 30 minutes, are much less likely to develop cognitive impairment as they get older. Is it complicated? Yes. It probably protects you from vascular disease of the brain. It releases all sorts of factors that are good for you. This is a new idea, exerkines. I love it. The idea is that when you are exercising, your body, your bones, your muscles, your brain are releasing all sorts of compounds, cytokines, things that prevent depression, prevent anxiety, prevent cognitive impairment. So I religiously preach to all of the people I see, whether they're healthy elders or even younger people, exercise must become part of your daily diet. Incredibly important. I just was going to compliment whoever designed the lunch for today, because this epitomizes what you should be eating. And I think diets that are heavy in meat and fats are associated with, no surprise, more likelihood that you will have cognitive impairment. Grains, leafy vegetables, Uh, uh, Particularly nuts, walnuts seem to be particularly valuable. Reduce the risk of stroke and dementia about 30%. Is it too late to change? We don't think so. So I think even if you've had an unhealthy diet for most of your life, I think changing it probably has some protective effects. Don't worry about the complicated slide here, but this is something that I just find absolutely amazing, and it's a very new finding. And this relates to sleep and developing degenerative diseases. So increasingly, we've realized from large studies, a woman's sleep study in particular, that women who do not sleep well longitudinally are at much higher risk for developing a degenerative disease. I believe the data that people who are on sleeping pills like benzodiazepine increase the risk for developing cognitive disorders as well. And now we're starting to understand the mechanism. And the mechanism is absolutely amazing. If you get into deep sleep, not dreaming sleep, but deep sleep, you activate a system in the brain that gets rid of the bad proteins. Contrary, if you don't get into deep sleep, those bad proteins that accumulate with Alzheimer's disease begin to accumulate. So this is the body's amazing mechanism to protect us against neurodegenerative diseases. So sleep well, think about the medicines that you take for sleep, but there's a methodology to this, and if you're not sleeping well, I think really needs to be addressed. We're getting some really good data that suggests if you combine these risk prevention strategies. This is the Finnish geriatric intervention study. There's studies like this going on now in Europe, the United States, again in South America, but suggest if you use these lifestyle measures that you will protect yourselves from neurodegenerative diseases. It's one of the most powerful and positive stories that I have heard in my lifetime in neurodegenerative diseases, and it's new, and I think it empowers people to do something about their own health. Just thinking very briefly about what you should think about if you're worried, and this is a program we have through the state at UCSF led by Howie Rosen that talks with people about the systematic way that doctors should approach your cognitive problems. The first is identify if there is a problem. If your doctor doesn't ask you, they'll never know. Do you have a cognitive impairment when we test you formally? We have all sorts of norms. We have all sorts of great ways of testing this. Do you have a treatable disease like poor sleep or hypothyroidism responsible for your trouble? What kind of cognitive impairment do you have? And then once we make a diagnosis, we're going to treat you because our treatments work. This is something that one of our young scientists, Kate Poseen, has developed. It's a tablet-based cognitive testing. It's short, 10 minutes. It's valid. It can detect cognitive issues. We're exporting this across the world to all sorts of different populations. And it's something I think that we're very proud of at UCSF, that we can do this very quickly and easily and help you determine whether you have significant cognitive issues. Dementia, is a broad term. It just means progressive decline in cognition. It can be memory, it can be visuospatial skills, it can be behavior, but it's a decline from baseline. And does it impair your day-to-day function? That's dementia. There are thousands of causes for dementia. So don't be satisfied hearing from your doctor you have dementia. What you want to hear is what kind of dementia. And this here shows some of the different dementias that people can suffer from. All different chemistry, all different clinical symptoms, and all different treatments. Okay, so here are the proteins that aggregate in the brain with these different degenerative diseases. I'm just going to say a few more words, and then I'm going to turn it over to Amy about the protein of Alzheimer's disease, which is amyloid beta and tau. So they're Taoists who believe tau is central to the disease. I'm a Taoist. They're amyloid beta people or Baptists who believe that the beta is responsible So, and the fervor of religious war is definitely present in our field. And then there are people like Amy who don't believe either one is really at the center of this. And these are the real innovators. But so I just show this as a shout out to Stan Prusner, who won the Nobel Prize, who set the stage for understanding degenerative diseases. He said, we can take a gene that causes a degenerative disease, put it in a mouse, in a fly, in a worm, and Amy has a massive cell culture system for different genetic mutations. Watch that disease develop, and Amy's going to talk about it from preclinical to clinical to dementia. And they're all caused by abnormal protein aggregation. Get rid of those bad proteins, we've got a cure for these diseases. Prevent them from happening, we've got a prevention. And that's where our field is going. This just shows you that we can now image the amyloid in the brain, and we can also image the tau in the brain. And so we can tell you 15 years before you're actually sick, whether that amyloid is present. We can tell you a few years before you're sick, because tau is so central to the sickness, whether tau is in the brain. This is a huge breakthrough. We can tell you you have early protein changes that make you very highly at risk for developing a degenerative disease. How can we get rid of those proteins? Even more remarkable, and our group has played a major role in this, we now have blood tests that capture those proteins, amyloid and tau. So the first one was FDA approved yesterday for amyloid. Pretty soon you're going to be able to walk into your doctor, and the doctor will be able to tell you, or your nurse practitioner, whether you're going to get Alzheimer's disease based on the presence of these proteins. It's not quite that simple, but it's almost that simple. It's a huge, huge breakthrough. Okay, this is some of our work. The blood tests correlate really brilliantly with the brain tests. So I think I'm going to stop there and just say that we're developing drugs for Alzheimer's disease. One of the questions is, can it clear the amyloid from the brain? We have drugs already, as shown here, that can clear amyloid from the brain. They haven't yet shown extraordinary efficacy in improving your symptoms longitudinally. So that's sort of where we are today. But there are enormous changes, and laboratories like Amy are developing smarter therapies that are going to lead to powerful preventions in our lifetime, I think in the next decade. So stay patient with us. Make sure that you're looking after your own health. And more importantly, if you or a loved one has symptoms, there are places to be seen. We'd love to see you at the Memory and Aging Center. So that's where we are today, but Amy's going to show you where we really are today, which is on the verge of treatments.
Usually I always have to lower those, but I'm mic'd up, so.
You want me to try?
No, it's fine.
Yeah.
Thank you, Bruce. So Bruce is always a really hard act to follow, and I will do my best. But first I want to say thank you to Joan, to Cheryl, to David, to all the organizing committee for having us here. It is a delight to be here, to interact with all of you all. My job is to, Bruce has already done a great job telling you about some of the things that you can do, probably already doing. And my job is to tell you kind of the basic science, the reasons why you're doing it. So we're going to be talking more from the point of view of a neuron, of a cell. And it's a real delight to be here at the Women's Health Symposium, because the last time I checked, I was a woman. And we've talked a lot about, the big M, memory. But, I'm someone who thinks about my own memory. I always am trying to think of that person's name. I'm not like Andy and Bruce. They're unbelievable. I remember people's names. I'm always trying to think of words, etc. I also think about the other big M. But I'm not going to talk about that because we actually don't understand that big M in menopause as well as we can in the future. So I'm going to be talking about how understanding the basic science of Alzheimer's disease might help us avoid it. Okay, So Bruce referred to and showed you pictures of these sticky proteins that accumulate in or around our neurons, our brain cells. Our neurons do everything for us. Every time I move, every time I blink or wink or think, it's a neuron firing in a very coordinated way. In order to stay coordinated, the neurons have to function optimally. And to do that, every one of our neurons is like a really complicated machine. There's tons of stuff going on, and it takes energy to do that, and it generates garbage. And so our neurons have to be able to clear out the garbage. And one of the problems with Alzheimer's disease is our neurons can't clear their garbage effectively anymore. And so it overflows and has to figure out where to put the garbage. One of the key organelle, really, the key part of our neurons, of our cells, that clears out the garbage is this little thing called the lysosome, okay? This is just a cartoon of it in the cell, but the lysosome actually takes all the extra garbage and chops it up into little pieces and sends it back out to the cell to reuse. It's actually kind of a recycling center for our garbage. And the lysosome works great for many, many, many years. But it becomes more and more challenged as we get a little bit older, and it becomes a little bit less efficient. So when that happens, the proteins in our cells, so our cells run on proteins, just like we do, and proteins are actually really interesting because we think of them as just something we eat or we don't eat when we're having. I had all-you-can-eat shabu shabu last night, and Andy is vegetarian. So, the proteins that we eat actually are structured, and we have to break them down in order to digest them. And the proteins that run our cells are incredibly complicated. They have to be folded in a certain way, almost like origami. Okay, this is a really simple origami. Our proteins are actually folded by our cells to do their function, and sometimes they get misfolded. And when we're young, these things are refolded and it's no problem. As we get older, we tend to clear the misfolded proteins, and that's what our lysosomes are doing for us. However, sometimes if the lysosome, and then the analogy is we have lots of proteins, and I don't know where this point is. If our proteins were a piece of paper, they need to be recycled, they need to be shredded, but if they aren't shredded, then they end up being trash that can't be cleared. And then our trash cans overflow. I'm not exactly sure where I'm supposed to be pointing this. Right here? That seemed to work. Okay, to the right? Okay, great, thank you. Oh, over there, great, thank you. I'll go back. It worked better when I put it in my head. Okay, and when the proteins are misfolded and they can't be cleared, then they end up in these in the amyloid plaques and tau inclusions, or in alpha-synuclein. Okay, so one of the things that we're beginning to appreciate more is that lysosomes, which are this recycling center, they're super acidic inside. Okay, I never thought about how there's like sort of acid within our cells. This is a pH scale, and it gives you a sense. Distilled water is a neutral pH of 7, but our lysosomes are really acidic. They're kind of like tomato juice and maybe even like orange juice in terms of how acidic they are. And they have to be super acidic in order to do their job. So here is the inside of a cell, and I'm going to highlight our lysosome right there. So its pH in this figure is about 4.7. When we've measured it in the lab with a tool that I'm going to tell you a little bit about, it's actually closer to 4. And so when the pH is normal, the lysosome can do its job just great. But as we get a little bit older, the pH is harder to maintain. And so it's starting to drift up, and the lysosome becomes less efficient. Okay, so lysosomes, we all want them to work perfectly, right, for the entirety of our lives. But we do know that with age, they become a little bit less efficient. And the things that make them less efficient with age include mutations, which hopefully none of us have, in certain genes that lead to autosomal dominant or inherited forms of Alzheimer's disease, Parkinson's disease, frontotemporal dementia. That's what those abbreviations are for, the underline. And the other thing that impairs function is age, but we don't completely understand yet why, although I'm gonna talk with you a little bit about what we think might help. But what we all wanna know is, how can we improve the function of our lysosomes, right? And that's what I'm gonna be giving you, hopefully, some tips about. And what we wanna do, if our lysosomes are, the analogy is that they're the paper shredder, we're gonna sharpen the blades and we're gonna pump up the motors. Okay, so the blade of the lysosome is something called a protease. So I told you that proteins are really complicated in their folding. This is an example of a relatively simple protein and how it needs to be folded to do its function. Proteases are the little machines within our lysosomes that are clipping our proteins from big pieces into little pieces. And when they don't work well, it's because they are inhibited by other proteins. So there's something called progranulin in our cells, and normally progranulin helps the proteases work, but as we get older, they're cleaved up into little pieces, into granulins, and they inhibit it. So one of the things we want to do is try and prevent progranulin being cleaved up into granulins, and that will help the lysosomal proteases work. And we're wondering if we can find drugs to do this. So one of the things my lab is doing is we're looking for drugs to prevent this process from happening. And then hopefully our lysosomes will work better. The other thing we can do is kind of soup up the engine. And so we want our lysosomal, our cute little beetles to be like, you know, the Bigfoot truck, okay? So how can we do that? Well, one of the ways we could do that is to inject a little bit more lemon juice into the gas tank. So I already told you the lysosomes need to be acidic. And so if we can develop medicines to keep the lysosomes acidic when we're older, then that might actually work. And by the way, I was the one that suggested, I don't know if anyone took my suggestion, the lemonade at lunch, but drink up. Yeah. Okay, so what is it actually about lemonade? Lemonade has tons of vitamin C, as do all these other foods. And vitamin C has another name. It's actually called ascorbic acid. It's an acid. Ascorbic acid likes to go to the lysosome and make it more acidic. And so if it doesn't have to be lemons, any of these foods, and I actually have more delicious looking pictures here, can help to keep your lysosomes optimally acidified and working most efficiently. And so Andy has it right. Fruits and vegetables, Bruce mentioned the Mediterranean diet. You all pick the perfect foods, but the more of these delicious looking vitamin C holders that you can take into your diet, the better off your lysosomes will be. Okay, there's a lot in the news now about intermittent fasting. And I will tell you that I actually tried to do this, but it's not actually easy. So intermittent fasting is typically, I looked on the internet, it actually says 16 hours, but the best data in the scientific literature is an 18-hour fast. But you only need to do it once a week to get effectiveness, okay? So if you can do an 18-hour fast, and that's like no calories, you can take water, you can take coffee, you can take tea. The reason it works is because it activates the lysosome, okay? So I do want to be clear, intermittent fasting, the goal from a neurological memory point of view is not weight loss. The goal is to maximize lysosome activity. During the fast, our cells get hungry and they say, well, there's no more food coming in through the stomach. So they start looking around and they actually start to engage a process known as autophagy, which in Latin translates to self-eating. So the cells actually start to break down their own components, which sounds like it's bad, But amazingly, the cells can figure out which parts of itself are damaged, and they actually clear out the damaged parts of the cells. So activating autophagy is good for us, and autophagy requires the lysosome. It is entirely dependent on the lysosome being optimally functional. Okay? And so it's like, you can think about it as like spring cleaning of our neurons. So Bruce already talked about exercise, and we know it's good for our heart, so it's good for our brains, but the exercise actually activates our lysosomes too. So if you can do a 16-hour fast with some exercise, that probably is as good as an 18-hour fast. Okay, there are certain cells that are vulnerable. We've talked about neurons some, and so we want to do everything we can to protect our vulnerable cells. And the way we do that is, like Bruce said, we need to socialize, to stay mentally and socially active, to promote the neurons being healthy. We need to sleep. Like Bruce said, our colleague at WashU has discovered that while we sleep, the neurons, as well as the neuron sister cells, the astrocytes and microglia in our brains, are taking extra garbage, and their lysosomes are optimally active to clear out these sticky proteins. And then one thing, that I learned from a colleague of mine is that actually there are particularly vulnerable cells called RPEs in our eyes. They are actually really busy clearing out garbage in our eyes. And so wearing shades is actually really important for eye health. So you look cool and you protect your eyes. Okay. And then, we all want to know what's good for us, and that's really, really important. We also, as you all are doing, want to stay engaged and active and supportive of the research that's going on, because it's the basic science research that kind of comes up with the new ideas that will lead to big breakthroughs. One example I want to give is, you all have all heard about these PCR tests that we all take now for COVID, right? PCR stands for polymerase chain reaction. It's something we do in the lab. The key part of it is something called Taq polymerase, okay? It's an enzyme that can copy DNA and RNA. And that Taq polymerase was discovered when people were down at the bottom of the ocean studying thermal vents. They're actually bacteria that live down there, and they can reproduce at 96 degrees. And that's why we have polymerase chain reaction with the ability to reproduce DNA and RNA at really high temperatures. That's fundamental to PCR. And it's because people were curious about, do bacteria live at the bottom of the ocean, and how? Okay, so this is my last point, which is that we are actually actively looking for ways in our lab to try and optimize lysosome function. And we developed a sensor that we call Firefly for fluorescent indicator reporting on pH of the lysosome. And with Firefly, which you can see kind of bouncing around, is a fluorescent protein that tells us, is the pH of the lysosome great or is it not great? With this, we can actually screen for drugs and medicines that will hopefully be able to use as an adjunctive for aging, for neurodegenerative disease, to keep our lysosomes optimally functioning. Okay, thank you very much.
All right. Thank you. Thanks so much to both Amy and Bruce. That was great, with the exception of the comments on my own lifestyle, but that was, we'll talk
Andy,
about,
I had one more.
we'll talk about that
Okay,
afterwards.
okay.
You hold on there, big guy.
Okay.
All right, so here's the deal. As always, a bunch of great questions. If you have other questions, pass them to the aisle. They'll be brought up this way. We've got a ton to go through, so let's just jump in. Bruce, let me start with you.
Yes.
You mentioned all these important, really good things that we can all do to be preventing dementia, but when I'm watching the Warriors game on TV, I see a bunch of ads for supplements I can get at the store. Why, exercise, that seems like a lot. Can I just take a pill? What is the efficacy of these things that we hear all the time that someone asks?
So it's an interesting story. It's a great question. The FDA has different rules for what you can say, whether it's considered a drug or a medicine or a food supplement. And so the rules are very loose about what you can say. I watch these ads, in shock, actually, because they talk about how great these supplements are, or whatever, proven to work. And actually, for some of them, they've been proven not to work. I mean, they have undergone systematic studies which show definitively, Prevagen is one example, sorry to the company, but proven not to work. So anyways, but now it's advertised on TV to work. What does it work for? I don't know. It works for somebody's wallet, but beyond that, it doesn't work.
So no supplements yet. Okay.
Yeah.
Amy, question for you. All these lifestyle interventions, we sort of portrayed them as something to prevent dementia.
Mhm.
If someone's starting to have cognitive decline, if somebody's in the middle, if maybe they've already had a diagnosis of dementia, can these work as well, or are we sort of too late in the lysosome or whatever pathway we're thinking about?
So I mentioned how complicated neurons themselves are. Our brains are even more complicated in the sense that the neurons have to be wired together in order to actually fully function. With dementia, we know that by the time we see symptoms, that there's already a fair amount of neuronal loss and neuronal damage. So right now, the state of the art is that we cannot reverse damage that's already been done. And that's I'm a big proponent of the growth mindset. I think those are our goals, to be able to rebuild circuits that are damaged. But regardless, none of the pharmaceutical agents will reverse anything. Even the best medicines we have don't reverse. After diagnosis, the goal is to slow the progression to zero, to keep people as healthy for as long as possible.
And then Amy, let me ask you sort of another one. You mentioned social interaction. What the heck does that mean? What should we practically be doing? I think it's great we're all together in person, for goodness sake. Maybe that counts. And then in the setting of the pandemic, when everybody's so isolated, are we seeing less of that, meaning more dementia? Talk a little bit about that story.
So I think Bruce and I have probably both noticed this with our clinics, that we had people who were doing well that we follow who said, I am much worse. Let me tell you all the things that have changed. That we heard a lot. Then we would test them, because we always take what people say to us really seriously. So then we would test them, and one of two things would happen. Either they were objectively the same and they were just feeling, because of the social isolation, that they were worse. Other times, and I'd say this was the minority, maybe 2 are fine, but sometimes people would actually be worse after being stable. And we think it's because of this lack of interactiveness. I don't know about you all, but when I'm sitting at home, even if I'm on Zoom every minute of every day, which I am, that there's just a limit to, even though I'm mentally active, there's not this sort of real engagement on a human level. And then I go to work and I see my colleagues and we talk about, the shabu shabu that we ate. And we talk about what our kids are doing. And we talk about, in a way that's spontaneous, that probably activates different parts of our brains. And importantly, brings out, I think, that emotional aspect, which is also chemical and is also good for our brains, in a way that we've been missing. So I do believe that that's a real phenomenon. Yeah,
I think that's helpful. Bruce, of the probably 100 questions that I have, 75% of them are about sleep. So let's dig into this a little bit more. So I'll start. Let me just pepper you with a few. One, does everybody have the same amount of sleep or deep sleep that they need? Does that change over a lifetime? And how long am I supposed to be asleep every night, Miller?
Yeah. Well, if you want to lead a healthy lifestyle, just follow Andy around. I mean, so he's got everything, better than the Mediterranean diet. I mean, it goes on and on. But the one area that he's kind of deficient, I think, is sleep. And but it also brings up the mystery of our own idiosyncrasies. And this is very new. This idea that deep sleep is a critical time for getting rid of toxins. I mean, this has just exploded in the last 5 years. So we don't know about individuals. I mean, there is huge human variability. We have great programs that Andy started at UCSF that study sleep. But what's really clear is if you do not get into deep sleep, that this is almost certainly a very significant risk factor.
And we know, just for everyone, that the amount of sleep that one, quote, needs changes as we get older. It gets shorter as we get older. So we have a lot of folks who come and see me in my clinic say, Doc, it's really tough for me to sleep, and this is extraordinarily frustrating. And a lot of times we just hear it's because they're used to 8 hours when they were in their 50s, and now it feels like after 6 hours they wake up early in the morning. That might be just fine for some people, depending on your needs. But Bruce, there's another piece that came up a number of times here. You said we needed sleep, but then there's this criticism that you leveled over some of the medicines that people take to try to get good sleep. So I think the audience, are we a little stuck here? If we're not sleeping enough and we take these supplements, but you said these supplements will give us some memory problems, what to do? Are there some supplements we should be taking? Melatonin, whatever? How do you resolve those two things?
Well, I mean, I think the one thing we know about the typical sleep medicines, like Valium would be an example, is that they increase your sleep, but they don't increase good sleep hygiene. And so one of the things that we lose out on is deep sleep. So you never get into that deep sleep state. We think that may be one of the reasons that studies of women in particular who took these medicines showed a higher rate of neurodegenerative disease longitudinally. I think we now understand it. It was just phenomena. Now we understand the mechanism. So there are certain medicines that you can use for sleep that don't affect deep sleep at all. Melatonin, as Andy mentioned, is always our #1 go-to. There's a certain kind of antidepressant called trazodone, which if I'm forced to using a sleep medicine is my preferred sleep medicine based on some really good data that's come out of our center. So I think this is a new science. I think we're going to learn a lot more about this over the next years. But, the hard billions of dollars of work that the pharmaceutical agencies put into figuring out ways of getting rid of amyloid, what they didn't realize is we had our own ways of getting rid of amyloid that were much more natural and brilliant. Yeah.
Amy, Bruce just hinted, and we've talked about it a couple times, women and an increased incidence of dementia. We know women live longer. We know that's for a variety of reasons, some of which is a healthier lifestyle. What is it about an increased prevalence of this disease amongst women? Is it just an epiphenomenon of folks living longer? Is it something about hormones and menopause? What do we know about this gender difference when it comes to the rates of these diseases?
I think there's still a little bit of controversy and disagreement around this, because I actually did look into the literature around the higher rate. And people have said it's not just because women live longer, but actually when I look at the literature, I'm still not 100% sure. So it is clear that estrogen is protective of the brain. That being said, there are many, many women doing just fine who are postmenopause, and the perimenopausal and menopausal symptoms actually do go away, right? So we can get to being, they're real, but after we're done, we can get to being just as sharp as we were beforehand. I would say that the science is still to be determined. I'm going to be digging into this a little bit myself, because I think it's actually really interesting. I would say there is a possibility, to go back to the sleep issue, that because sleep is very disturbed, perimenopausally oftentimes and postmenopausally, that may be actually a contributor. And I guess I'd love to weigh in a little bit on the sleep in general, because we actually have 2 world-class sleep scientists at UCSF, Louis Potacek and Yingwei Fu, and they have studied the genetics of sleep. And there are people who are, early birds, and there are genes responsible for people who are early birds. And then there are night owls, and there are other genes responsible for night owls. Then there's this thing called congenitally short sleepers, people who literally only need 3 or 4 hours of sleep a night. That would be Dr. Andy Josephson. And these people sleep 3 or 4 hours a night, but they're awake, they're energetic. They oftentimes, the social aspects, they oftentimes work multiple jobs. Andy has at least 10. And they stay cognitively sharp for a really long time. So what I'm just trying to say is that there's a lot of inter-individual variability. And what we're seeing now are the means, but I think for any individual, just like with actually neurodegenerative disease, we have to understand the personal sort of genetic, social, and other aspects of the person to understand. So I guess I don't want to strike fear into people's heart that because my mom is always asking me, she calls me Mimi, I can't sleep, am I going to get dementia? And I say, it's okay, Mom, it's okay, you're actually really sharp. You know, we can do the mini mental status test again, every individual is different. And the other things that help sleep a lot actually are exercising, eating right, avoiding too much alcohol late at night because that disrupts sleep, eye shades, no blue light, all things that you all know. But sleep hygiene is actually really important as well.
Bruce, maybe the biggest reaction I sort of heard people talking to each other about is when you mentioned that we've now got a blood test after a PET scan, but now we've just got a blood test that can tell us if in 10, 15 years we're going to develop Alzheimer's disease. Wow. So practically, why would we want to get one of these now? And I think with all seriousness, how does this set us up for trials of medicines, and maybe it teaches us a little bit why some of these trials of medications for Alzheimer's have failed.
I love Amy's comments about precision, and I think that's what we're trying to do in our memory clinics at UCSF. Each one of you has different biology, different genes that increase your risk, decrease your risk. For degenerative diseases. Some things that we don't understand yet may trigger a degenerative disease process. So I think the idea that all we had to do was get rid of amyloid in the brain and we would cure all these diseases was really mistaken. And it was a really bad strategy. And in the early studies, we learned that people who had antibodies against amyloid, a third of the people in the studies didn't have amyloid in the brain. So, I mean, the studies were just very badly done, and that's changed. So we are now approaching a precision, and we didn't even touch on some of the other markers that I think we're going to be able to tell you about, like the number of synaptic connections, the health of your neurons in your brain. These are all things that are coming out of science that will allow us to, in a very precise way, look at an individual in our clinic, know their susceptibilities. And then going back to Andy's question, why would you want to know? Well, honestly, I wouldn't want to know in 2022 because it won't change anything other than increase my worry a little bit about it. But in 2025, you may want to know that you're accumulating amyloid. Even a denier like me might want to know because I would know there are some new therapies.
So right now, these tests can be used if someone's having cognitive complaints
exactly.
to understand what the actual diagnosis is.
Exactly.
But figuring it out 10 years ahead of time, right now, that's not for us, but may be important for these clinical trials. And of course, in the Memory and Aging Center, we have 1 million clinical trials going on at any one time for these diseases. Got it. Amy, you mentioned your mom. So let's bring her into this. Someone asked, what advice do you have if we have elderly friends or parents who are developing some cognitive problems? Should they be evaluated? And perhaps, you're such a great clinician, Amy, perhaps a little bit more on sort of what constitutes something to worry about versus just, well, of course, they're X years old. We would expect things to not be as sharp as when they were 20. Help us out with that a little bit.
I mean, it's really tough, and I really am personally struggling with this right now with elderly parents. So what we do know, I'm going to take a step back, is that people in supportive environments are going to do great. But unfortunately, in our society, oftentimes there are people trying to hack your email and get you to pay this, that, or the other. And so there's some bad actors out there. And so we need to protect our friends and family, right? This can be tough because oftentimes when people start to have symptoms, it's part of the illness actually. They lose insight. There's a part of our brains that gives us insight into our own limitations, and that's affected in these illnesses. So, you know, somebody could, you know, be giving away thousands of dollars. I am speaking from personal experience, to you know, people who are trying to take advantage of them and not actually realize that this is bad. And that's really tough. It's really tough on friends and family, right? So I think once again, every individual is different, and handling it is going to have to be different. Sometimes it is best to say, I'm worried about you, and maybe we should think about getting an evaluation. That is very reasonable. Other times, if it's really tough, then actually going a little circuitous route to protect people, to get them sort of evaluated, and you know, financially protected, that's okay. I think families feel really bad about that, but sometimes that is in that individual's best interest.
And it's common when folks are evaluated at the Memory and Aging Clinic, you know, one of the more common outcomes is a diagnosis of everything's okay.
Yeah.
And we're happy to do those evaluations as well. Bruce mentioned a cohort of healthy aging patients that we follow longitudinally who get a bunch of tests, and we try to figure out, well, these are great agers. How are they doing this so well? That's a fine outcome as well. So I think, as Amy said, if one is concerned, trying to get somebody evaluated makes a lot of sense.
And I will say that sometimes an informal evaluation can help. I mean, I think Bruce and I, we've seen enough that we do know this is something to worry about, or this is not something to worry about. This is okay. Yeah.
We've got time for just a couple more before we have to end. Bruce, you know, I think we all love coming up here for a variety of reasons, but one is, you know, it's wine country, and you're sitting here giving a slide where you say excessive alcohol is a problem. So somebody asked, can you define what we mean by excessive alcohol? How about, you know, let's not ruin an entire evening here. What do you mean, Bruce? When, how much, what's okay, and what do you get concerned might be contributing to cognitive problems?
I think for all of you and for me, sometimes epidemiology is frustrating. So, you know, one week you'll hear, you know, 2 to 3 drinks a week is good for your brain. And these were good studies. I wasn't sure it was correct, but these were good studies. The trend has been a little bit more negative. I think, again, people are so individually different. You know, I think for me, 2 or 3 drinks a week is fine. I think often we know when alcohol is a problem for a loved one, you know, and it's, you know, half a bottle of wine or more. Oh, boy, I'm going out on a limb here.
Here we go. Let's go.
Half a bottle of wine or more a night, that seems, considering the epidemiologic data that we have, that seems like a lot. So anyways, that's as close as I dare come. Yeah.
Many of you, I have a lot of friends in the audience. Many of you know I'm a stroke doctor, and that's what I spend a lot of my time doing. I'll tell you, with stroke, it's a very similar relationship, that we know that actually probably some alcohol a week, not being a teetotaler, is good for cardiovascular and cerebrovascular health. And when you get to more excessive amounts, like Bruce was describing, then the rate of stroke actually goes up. So finding that sweet spot where enjoying some alcohol but not too much ends up being important. I know you want an exact amount. We just don't have the science to tell us that. I want to finish with something fun. I want you to each just take a minute in closing and tell us where you think this field's going to be in a few years, or what's exciting to you. Amy, you're on the cutting edge. You're one of these physician scientists nationally who everyone looks to for what's going to be the next big thing. You got a chance to tell us about your lysosomal work. By the way, that lysosome story, that's pure Gal Lab. I mean, Amy is the world's leader in thinking about that. And it may very well turn out to be a concept that is revolutionary in terms of how we treat and think about dementia. But Amy, I'll let Bruce go second. You tell us, where are we going? What's exciting in this field coming up?
I think there's a lot to be excited about. We're kind of zeroing in on, for each individual person, right? What are the aspects of lysosomal biology that could be different. And so we may, this will require things like everyone getting their whole genome sequenced, and then us looking at specific aspects of the genome to figure out if there's something that we can intervene on. So I think really exciting is the precision, the personalized approaches to neurodegenerative diseases. And, the drug screening efforts, they're really exciting. They do take a while. We can do them in the lab, and then even once we get a drug, it's another 3 years, maybe more, to get them through the FDA. But there are these things that we know, like vitamin C, that we can kind of do right now that can potentially help us. But I think the future is precision medicine, not around the aggregates that we see, which I think of as the result of the disease. We want to go upstream. We want to get the inciting events. We want to intervene early and prevent the diseases, because it's going to be longer for us to reverse them.
It's great. Bruce, final word for today. Yeah. Where do you think we're going? What's exciting for you over the next few years?
I would sort of echo in a slightly different way what Amy said. I think we can't, at the Memory and Aging Center, do all the screening in California. Your healthcare system has to do it. But I think what will happen is the screening will become routine. The brain is every bit as important as any other organ. And so it's, I think, strange that we aren't screening elders for a system that really is important to our well-being in every possible way. So I think early screening for how we're doing, painless. I think we're getting better at this. Early screening, I won't be surprised if every bad neurodegenerative disease, before you get it, can be screened in your blood. So screening for risk, just like we look at risk for a stroke. Interventions, slowly, but one by one. I am sure that one of Amy's compounds that work on the lysosome will become, for us, like taking a cholesterol drug. So I think, and slowly increasing the quality of life as we age, because the brain is so strongly tied to it. And last thing we haven't touched on today, which is caregiving. And I think many of you as daughters or wives are in the midst of caregiving at every possible level of generation. And having systems that support you as caregivers is unbelievably important and will become part of our society as we move forward. Kate Poseen at our site has shown that supporting caregivers is cost effective. It decreases the likelihood that someone will end up in an emergency room. It increases quality of life for the family. So this has got to happen in our society. So, yeah.
Dr. Gao, Dr. Miller, thank you so much. Wonderful to have you here with us today, sharing your wisdom. We really appreciate it. Thank you.
Thank you.
And to Dr. Josephson, we couldn't do it without you. Thank you for bringing it all together. We have some gifts for you. And as you would expect in wine country, it is going to include some wine.
Thank you so much.
Thank you very much.
Wow.
Wow.
Yeah, heavy. I won't drink it all tonight.
Just don't drink it all at once.
Yeah. I'm not drinking more than a quarter of the bottle tonight. Yeah.
Wow.
Great. So I'm Dave Fear. I'm the executive director of the Hospital Foundation, and we're just so pleased to have you all here today. Thank you for being with us. We did all the fundraising for this event on the front end, and that was intentional so that we could focus on the educational aspect of the day here. And yet the fundraising is still super important, right? It helps us be able to do amazing things for the hospital. And so today, all of our patrons, our sponsors, and all of our guests, you all have helped generate a lot of funds, and we've been able to purchase a new ultrasound machine for the hospital. And as Sherry mentioned earlier, we are able to continue the Project Pink program because of your support. So thank you very much. Really appreciate it. I want to call out a few of our key sponsors and patrons. So our lead presenting sponsors are UCSF Health. We really appreciate their support, and Union Bank as well. So yeah. Our angel patrons include Joan and Sandy Weil, Kimberly Blattner, Sherry Hughes, the DeBalman McGrain Group, Jamel and Tom Perkins, Elizabeth Seeley and Peter Seeley, and the Videz Family Foundation. So thank you to all of you. And a shout out to the Haystack Farms and the Wildflower Garden Club. So all of the beautiful flowers that you see around here, that's because they put those together out of their gardens. So thank you so much for Kimberly for organizing that as well. So please stay to join us outdoors. I know it's warm. There's some shady spots. We have wine and dessert and a little bit of social time. And the docs have said that they would stay and mingle. So if you have any further questions, I'm sure they'd be happy to chat with you out there. Thank you so much, and we hope to see you next year. Bye-bye.
Okay.